In September 2007, a spacecraft carried 2 species of microscopic animals into low Earth orbit. Then a tray opened and exposed them directly to space. No air, almost no pressure, cosmic radiation, and, for some groups, unfiltered sunlight. 10 days later, the tray closed, the capsule returned to Earth, and scientists added water. Many of the tiny animals began moving again. Tardigrades had become the first animals shown to survive direct exposure to space. But the famous headline hides the most important detail. Space is therefore true only with conditions attached. Vacuum was surprisingly manageable for the dried animals. Direct sunlight was a much harsher enemy. Why can drying help with radiation? Desiccation and radiation both create severe molecular damage, including broken DNA and oxidative stress. Adaptations that evolved for repeated drought can also help an animal tolerate other insults. Some tardigrades make a protein nicknamed Dsup, short for damage suppressor, which associates with DNA and can reduce certain kinds of damage. In laboratory experiments, putting a Dsup gene into cultured human cells improved their resistance to X-rays. But Dsup is not a universal force field. It was identified in particular tardigrades. Not every species uses the same molecular toolkit, and radiation tolerance involves protection, antioxidants, and repair working together. Scientists are still mapping which genes matter under which conditions. NASA's Cell Science 04 experiment later sent living Hypsibius exemplaris to the International Space Station to study short-term and multi-generational responses to spaceflight, including which genes switch on and off. The goal is not to turn astronauts into water bears. Tardigrade biology could inspire better ways to preserve medicines, food, vaccines, cells, or other biological materials during long missions. If researchers understand how proteins and membranes remain stable through drying and radiation, parts of that strategy might be borrowed without copying the whole animal. Tardigrades also matter to planetary protection. A hardy animal surviving a short orbital exposure does not prove that it could travel alive between planets. Years of radiation, extreme temperature cycles, impact shock, and the need to revive in a suitable habitat create much harder barriers. In 2019, an Israeli lunar lander called Beresheet crashed while carrying dehydrated tardigrades sealed into a sample. There is no evidence they woke up, reproduced, or colonized the moon. Without liquid water, food, and a protective environment, a ton is only a dormant passenger, and probably a damaged one. They are not immortal on Earth either. Active tardigrades can be killed by heat, toxins, predators, or simply the wrong conditions. Even dormant tons have limits that depend on temperature, exposure time, humidity, species, and how successfully they dried. Spectacular laboratory survival after brief exposure is often turned into a claim of permanent invulnerability. Duration matters. A few minutes at an extreme temperature is not the same as days, and surviving is not the same as remaining fertile and healthy. This is what makes the Foton-M3 result more impressive, not less. The researchers built proper control groups, separated vacuum from ultraviolet exposure, returned the samples, added water, and tested whether the animals actually recovered and reproduced. The experiment did not discover a creature that ignores physics. It revealed an animal whose drought adaptations accidentally prepared it for conditions evolution had never encountered above Earth. So, did tardigrades survive open space? Yes, dehydrated specimens survived about 10 days of direct space vacuum, and some even recovered after combined exposure to vacuum and solar radiation. But their greatest power was not toughness in the ordinary sense. It was the ability to shut down, protect a fragile biological structure, wait, and restart when water returned. The first animal to survive open space did it by becoming, for a while, almost indistinguishable from something that was not alive at all.